Howdy folks, welcome to part 2 of the Chapter 9 Notes, Molecular Genetics. In the last video, we talked about the structure of DNA and how it was discovered that DNA was the genetic material. In this part, we're going to talk about the semi-conservative replication of DNA. Now keep in mind that semi-conservative replication is the idea or the way that DNA replicates in which each parental strand is a parent for a new strand. So let's say you have some, quote, old DNA or original DNA. What happens is that when a new strand is made, it is going to be made in what is called a semi-conservative manner. In other words, the original strand acts as a template for the new strand, which will be drawn in red. Now, the other two theories that had been thought of as the method of replication had been one as known as the conservative and dispersive replication. But it was Meselson and Stahl who definitively showed that its semi-conservative replication is the method of how DNA replicates. So when we look at the big, broad overview of DNA replication, the first part is going to be opening up that DNA and making it available for the enzymes to copy the DNA. Then the second part is the actual copying and connecting of the base pairs. So we're going to look at each of these parts separately. Now, one of the big problems with that these enzymes are going to have is the fact that the DNA is protected with this double strand. So the backbone is not going to be the genetic information. What we're going to need to do is we're going to need to come in and separate the two strands represented with these dots here, which are the hydrogen bonds. We're going to need to be able to split that so that the enzyme machinery can come in and read and copy the DNA. Now, keep in mind that when that happens, that it's going to copy the 5' to 3' end, and then it will also copy the 3' to 5' end, and that's how we're going to get two copies of the DNA. So during DNA synthesis, the new nucleotides are added to the 3' end of a new strand, which has a free hydroxyl group. Remember, when we're looking at the deoxyribose sugar, we start from the oxygen, and from there we label the carbons clockwise. So the hydroxyl group that we are going to be added to is the one that's found at the 3' end. In other words, at this carbon right here, this 3' carbon, there will be a hydroxyl group. And this is where the enzyme is going to come in and remove this hydrogen here to form a phosphodiester bond between this soon-to-be nucleotide and the next nucleotide. And this is going to play a very important part in our discussions when we have to replicate both strands. So DNA is synthesized from deoxyribonucleoside triphosphates, DNTPs, or just think of them as deoxyribonucleotides. During synthesis, two of the phosphate groups are released, and the final nucleotide is a monophosphate, and it'll still have adenine, thymine, cytosine, or guanine, and the release of the other two phosphate groups provides energy for the formation of a phosphodiester bond. DNA replication begins with the binding of a large protein complex, which is known as a pre-replication complex, to a specific site on the DNA molecule called the origin of replication. The complex contains DNA polymerase, which actually adds the nucleotides. So starting with this origin of replication, which helps the machinery and enzymes and proteins know where to start the replication process, we're going to have to open that DNA strand. We're going to have to separate the two strands and allow for the enzymes or that complex to come in and start reading and adding nucleotides. In prokaryotes, the pre-replication complex binds to the ORI, the origin of replication. The DNA unwinds and the replication proceeds in two directions. Now, the replication fork is the site where the DNA unwinds, and that's thanks to an enzyme that's known as helicase. So this diagram is giving you a summary of the difference between how DNA replication proceeds in prokaryotic cells, as you can see here with the circular DNA. You can see the replication bubble here is getting larger and larger as the DNA is being copied. But in eukaryotic cells, because because it's linear, it would take a lot of time. So we have multiple origins of replication forming multiple bubbles, as you can see here. And then the bubbles travel towards each other. And so right here, specifically, and right here, those are the replication forks. Eukaryotic chromosomes are much longer and linear and will have multiple origins of replication, which speed up replication. It would take weeks to fully replicate a chromosome from a single origin of replication. That's why eukaryotic cells have many. DNA replications begins with a short primer or starter strand, usually a short single strand of RNA. The primer is complementary to the DNA template and is synthesized by a primase. DNA polymerase then adds nucleotides to the 3' end of the primer and continues until replication of that section is completed. So you can see here is the primer laying down this RNA primer in green. Once the primase has laid down a primer, now DNA polymerase 3 can come in and begin actually building new nucleotides to this strand. So you can see here's that short primer, and in here in red is the new strand of actual DNA that's being laid down by DNA polymerase 3. DNA polymerases are very large and shaped like an open right hand. The palm brings the active site and substrates into contact, and the fingers recognize the nucleotide bases. The enzyme then changes shape and catalyzes the formation of a new phosphodiester bond. A replication fork opens up in one direction. The two DNA strands are anti-parallel, with the 3' end of one strand is paired with the 5' end of the other. DNA replicates in overall the 5 to 3 direction. The leading strand is oriented to grow continuously at its 3' end as the fork opens. The lagging strand is oriented so that the exposed 3' end gets farther from the fork. Synthesis of the lagging strand occurs in small discontinuous stretches called Okazaki fragments. And so you can see here is the primer and DNA being replicated towards the replication fork, getting longer and longer and longer. But on the opposite strand, what you're going to have is the primer laid down ahead of the actual DNA strand, and the DNA strand is going to be made backwards. But then what happens once it gets to the end, another primer will be laid further ahead of the strand, and then DNA polymerase 3 can then come back further and work in this direction. Again, this is all because of the need of that free 3' carbon to attach the next nucleotide to. And so because we're having to kind of make a segment here, jump backwards, make a segment here, jump backwards, and continue making segments this way, they are known as Okazaki fragments. Each Okazaki fragment requires its own primer. DNA polymerase adds nucleotides to the 3' end until reaching the primer of the previous fragment. A different DNA polymerase then replaces primer with DNA, that would be DNA polymerase 1, and then the phosphodiester linkage between the fragments is catalyzed by the enzyme DNA ligase. So the primer is needed only once on the leading strand, but you will need multiple primers on the lagging strand. DNA polymerase works very fast but makes very few errors. This is called processive. It catalyzes many sequential polymerization reactions each time it binds to DNA. A DNA polymerase can add thousands of nucleotides before it detaches from DNA. When the last primer is removed, no DNA synthesis occurs because there's no 3' end to extend. A single-stranded bit of DNA is left at each end. These are cut and the chromosome is slightly shortened after each replication. So you can see here how there is a small amount of DNA that is left over. That's what's known as the telomeres. So at the telomeres, what's going to happen is this small bit of DNA is left over, and so it's going to get cut off. Now, if that happens continuously, every time a cell divides, a little bit of the end gets cut off, a little bit at the end gets cut off. Eventually, you're going to run out of the DNA. So at the ends is pretty much just nonsense DNA that acts as a buffer. And so the longer your telomeres are, the more replications your cells can go through. There are some cells that have an enzyme that are known as telomerase that will go in and fix the ends, and so that you don't get chopping off of the ends at every cycle. These are mostly your germline cells. Chromosome ends must be protected from being joined with other chromosomes by the DNA repair system. Telomeres are repetitive sequences at the ends of eukaryotic chromosomes. The repeats bind a protein complex called shelterin, which protects the ends from being joined together. The repeats also form loops, which are also protective. So you can see how gametes and some cancer cells have that enzyme to protect the ends of the telomeres. After 20 to 30 cell divisions, the chromosome ends become too short and the chromosomes lose their integrity, and apoptosis ensues. But continuously dividing cells like bone marrow and gametes maintain their telomeric DNA. Telomerase is the enzyme that catalyzes the addition of lost telomeric sequences. It has an RNA sequence that acts as a template for the telomeric DNA. Telomere lengths tend to shorten with aging. If a gene expressing high levels of telomerase is added to human cells in culture, the telomeres do not shorten and the cells become immortal. This is also seen in mice that overexpress telomerase. They live longer. Cancer cells also express telomerase. DNA polymerases can make mistakes in replication, but most errors are repaired. The two major repairing mechanisms are proofreading, so DNA polymerase has a proofreading function, and if bases are paired incorrectly, the nucleotide is removed. There is also mismatch repair. After replication, other proteins scan for the mismatch bases missed in the proofreading and replace them with the correct ones. And so here you see diagrams of DNA polymerase proofreading as it's creating the new nucleotides, and then mismatch repair, which would come in and remove that section and replace it with the correct nucleotide sequence. Hope this was helpful. Thanks for watching, folks.